My name is Kory Lavine. I'm an associate professor at Washington University in St. Louis, and it was my pleasure to be one of the co-moderators for the session on inflammation and HFpEF.
And I'm Julie Bossuyt from the University of California, Davis. We just wrapped up this session and we had some very interesting science on inflammation in heart failure and new HFpEF models. So, Kory, what did you think about the inflammation session?
Thank you. I thought it was outstanding. We learned a lot of new things about neutrophils, macrophages, and T cells within the heart. We learned from a talk by Albert Dada that neutrophils are recruited to the heart through two individual waves. There's a first wave that's regulated by demargination from the blood vessel wall within the first 24 hours, and then a separate pool that comes from hematopoietic tissues like the spleen and the bone marrow. And he discovered that the first wave is deleterious and regulated by catecholamines, suggesting some novel therapeutic insights. We also learned from Gopal Krishna Sharit at Ohio State University that the second wave of neutrophils, which is regulated by granulopoiesis, is regulated by netosis. And he nicely showed through genetic means that through netosis, neutrophils release S100A8 and A9, which signal through TLR4 and regulate granulopoiesis through IL-1 beta. In a very interesting talk by Roshwi Lee from Albert Einstein College of Medicine, who focused on the role of integrin alpha 5 in macrophages, showed us that deletion of integrin alpha 5 using two independent strategies to remove that gene from macrophages led to exaggerated LV remodeling after myocardial infarction, and that integrin alpha 5 was important for regulating VEGF and CXCL1 as well as CXCL2 expression, which may play a role in angiogenesis. Jesus Jimenez from Washington University provide really interesting information that CD40 agonism, which is a new target for checkpoint mediators in oncology, actually reshapes the cardiac immune landscape by potentially signaling to CCR2 positive macrophages and leading to the emergence of pathogenic effector T cells. And continuing on the T cell trend, Sahil Gupta from Ohio State really took a very deep and depth transcriptional dive into CD4 T cells and looked at two pools, one within the heart and one within mediastinal lymph nodes. 8 weeks following MI showed that these pools of T cells actually have very unique transcriptional signatures, and that CD4 T cells within the heart of MI animals are much different than those of naive animals. These findings are quite interesting and really open the door to understanding more about T cell heterogeneity, diversity, and trafficking in chronic heart failure. Julie, I'll send it back to you for what we learned about HFpEF.
Yeah, so before we get to the HFpEF, we actually had a very interesting study on the role of interleukin 10. And so they looked at the role of impaired intestinal permeability and severity of Gary disease that you find in basically late phase of a tech model. So previously people had found that in the early phase it was important, but in this case, interleukin 10 limited intestinal permeability and systemic levels of the peptidoglycan, which would otherwise induce inflammation and mitochondrial dysfunction. This gut cardiac axis was also explored in a pre-HFpEF obesity model of heart failure dysfunction from the GAO group and so they found using fecal matter transfer studies that butyrate played a key role in metabolic homeostasis and LRP3 activation. Of note, establishing the valid preclinical models of HFpEF is absolutely critical in that context. So very few models have been able to recapitulate all of the clinical features. And so in this new model from the UCLA group, they used basically a DBDB mouse with superimposed on that a very short-term TAC stress where basically you have a hypertrophy of about 1 week. So in this novel HFpEF model, what they found is that based on RNA-seq, is that there's a critical role for an alteration in crosstalk signaling between the glucogen and insulin signaling. So, but it's a very intriguing new HFpEF model, which might be useful for later studies. So finally, we had a very intriguing alternate model that looks at basically cardiac laminopathies, where they found a link between nuclear nuclear destruction and cytosolic inflammation. So here they found that the nuclear rupture induced basically a release of a protein HMGB1, which is a potent pro-inflammatory protein, which is normally sequestered by interacting directly with laminin C in the nucleus, but which is released upon basically laminin AC production and expression. So this basically links very intriguingly nuclear inflammation with basically overall cardiac dysfunction. So overall, very exciting information on inflammation. What did you think, Kory?
I really enjoyed our session. I learned a lot about all the different immune cells that live within and outside the heart, as well as new and emerging models of HFpEF. We had a great Q&A session on the different HFpEF models, including the new model that you mentioned, as well as high-fat feeding and other diet-induced models that have been used previously. We had a great back and forth amongst the panelists about the upsides and downsides of each model and experimental approach, which was great to hear and a great discussion to have.